All right. Good morning, everyone, and thank you for joining us for today's Winship Grand Rounds. If you're an Emory University or Emory Healthcare employee and you would like to receive CME credit for attending today, the login information can be found in the chat feature at the bottom of your screen. If you have any issues with this webinar or with the CME login, please send Kadiata Fofana an email or drop a note via the chat feature. This morning, we are pleased to welcome Dr. Christopher Klebanoff. Dr. Klebanoff completed his bachelor's in chemistry at Emory University in 2000. After this, he went on to receive his MD, also from Emory University, so he's one of our own, in 2006. And he is a medical oncologist and an NCI Merit Award funded physician scientist who has more than 20 years experience in both the preclinical and the clinical development of T-cell based immunotherapies for treating cancer. He is currently an assistant professor of medicine at Weill Cornell Medical College and an assistant member at Memorial Sloan Kettering, as well as an assistant attending physician at Memorial Hospital for Cancer and Allied Diseases. Prior to joining Memorial, he was an assistant clinical investigator and a Howard Hughes Medical Institute research scholar at the National Institutes of Health in Bethesda, Maryland. And as a member of the NCI surgery branch's senior staff, where he spent a number of years, he contributed to the early phase clinical development of a number of exciting T-cell based therapies that many of us know and love today, including the anti-CD19 28 zeta CAR that would later become Yescarta, and the gene engineered TCR therapies targeting a number of antigens, including NY-ESO, MAGE family proteins, and HPV E6, along with other TIL therapies. So really a pioneer in this space and has done amazing work surrounding cellular therapy and cancer. So he really pioneered the paradigm that T-cell subsets with memory-like attributes are critical in determining efficacy of adoptive immunotherapy. Furthermore, he resolved how host lymphodepletion, a standard practice that's used across cell therapy administration to patients, can enhance the potency of adoptively transferred T cells by removing sort of these homeostatic cytokine sinks. And currently, the Klebanoff laboratory is focused on discovery and immune targeting of these shared or public neoantigens that result from recurrently mutated proteins. These are really important in the pathogenesis of cancer and maybe immunologic targets. So in my opinion, after being in the field of immunotherapy for many years, this could really be a game-changing series of discoveries that launches therapies, and we'll hear about that today. So over the years, Dr. Klebanoff received numerous awards and honors, including the Merit Award I mentioned. He was a Damon Runyon clinical investigator. He has many publications in remarkably high impact journals, is well funded and just simply doing game-changing research. So we're really thrilled to have him here today with us for Grand Rounds, and welcome, Dr. Klebanoff. I'll turn it over to you.
Greg, thank you so much. It's a real honor to return to Emory in this virtual format. So as Greg mentioned, what I'm going to talk to you today is the efforts in my lab over the last 5 years to discover, characterize, and therapeutically target a new class of shared neoantigens, which we've termed public neoantigens. And I'll describe in a couple of slides a little more about what we mean when we say public neoantigens. As part of today's lecture, a couple of relevant disclosures. I'm going to talk about a number of different TCR gene sequences that target these shared or public neoantigens, for which my home institution, Memorial Sloan Kettering, has filed provisional patent protection for. So we're going to cover 3 different topics today. The first is I'm going to give an overview of both the unique opportunities as well as some of the unique challenges of using an evolving class of antigen receptors, the T-cell receptor, as a form of cancer treatment. Next, I'm going to give a summary of a paper that we recently published, which is our first example of the capability and possibility of targeting shared neoantigens. And this is a case study where we resolved that a commonly mutated driver, PI3 kinase, can give rise to a shared neoantigen that we can therapeutically target. And I think the takeaway message from this particular story is that TCRs, like so many other aspects of biology, are in a place where the structure of the molecule can dictate potency and safety. And finally, I'm going to conclude with what is presently unpublished data extending our efforts into targeting public neoantigens for a commonly mutated driver, the NRAS Q61 mutation, which is associated with cancers of high unmet medical need. And the takeaway message from this particular story is that TCR cross-reactivity, which is really a feature of all TCRs, a parameter that's sometimes referred to as TCR degeneracy, we often think about in a negative light, but in this case, this is a really interesting example where cross-reactivity can actually play in our favor, as I'll show you. So by means of background, I think our entire field owes a tremendous debt of gratitude to chimeric antigen receptors, or CAR-modified T cells, which have now been FDA approved since 2016 with, in total, 6 different unique CARs for hematologic indications. CARs make use of, in most cases, the variable domain from a monoclonal antibody that has been constructed genetically into a single chain format, grafted onto a stalk, along with some intracellular T cell activation domains. And this has really been a game changer for, in particular, B lymphoid malignancies, with durable responses seen in patients who have had otherwise refractory diseases. Now, despite these virtues, CARs do have one major limitation, which extends from the fact that the antigen binding domain of a CAR is derived from a monoclonal antibody. And that is, like antibodies, CARs can only recognize cell surface associated structures. So when you view this through the lens of the landscape of potential targets, this means that only perhaps 11% of the cancer proteome is potentially accessible or therapeutically amenable to a CAR-based therapy. This is a major barrier, in particular when we go beyond hematologic malignancies to solid cancers, where there's really a paucity of cancer-specific targets that we can target and have a minimal risk of on-target toxicity. Now, whereas CARs can only recognize cell surface molecules, an other immune recognition molecule, the natural T cell receptor, can recognize molecules both that are on originating from the cell surface, as well as proteins that are contained within the cytoplasmic, as well as intranuclear space. And the way that the TCR can accomplish this, I'm a little bit biased, but I think is one of the greatest magic tricks in all of biology. And we'll talk a little more about how this trick can occur, that an immune receptor can look at the intracellular proteome. Now, in doing so, of course, the TCR has access to the 89% of protein-derived antigens that are contained within a cell. And this gives us access to targets that are not just tissue differentiation antigens, which are analogous to CD19 or BCMA, but cancer antigens that are far more cancer selective or even cancer specific. So examples of these particular antigens can include cancer germline antigens such as NY-ESO-1 or MAGE, for which there are late stage clinical trials in the TCR space, in particular for a subset of sarcomas. Epitopes derived from transforming viruses, such as the human papillomavirus, analogous to work that Hayden and others are doing at Emory. And finally, 2 different classes of antigens that result from the mutational processes that give rise to cancer, private and public neoantigens, which I'll describe in the next slide. So let's talk about how this magic trick, the ability to recognize antigens that are within a cell, actually takes place. So this was Nobel Prize winning work done by Doherty and Zinkernagel. And so the trick of this particular magic trick is that the TCR doesn't actually directly look inside of another cell, but rather it recognizes a small proteolytically degraded peptide fragment that has been degraded within the proteasome inside a cell, loaded into a molecular basket-turned human HLA, and then finally shuttled and displayed to the cell surface. So why aren't we using TCRs more frequently? Well, there is never a free lunch in biology. There's never a free lunch in oncology. So it is this HLA dependency of a TCR that adds an extra layer of complexity. CARs, because they recognize the antigen itself in a way that is unprocessed, do not require HLA. But TCRs do require HLA. And part of the reason that this is non-trivial is that the genetic locus that encodes the human leukocyte antigens, this small region of chromosome 6p, the P standing for petite, is the one region of the human genome that is the most polymorphic. It is the most distinguishing genetic feature of all human beings. And so a TCR therapy has to align patients to not just one, but two different biomarkers, the target antigen and the specific restricting HLA allele, which again is the most polymorphic region of the human genome. So I've mentioned a couple of times now, private versus public neoantigens. What do I mean when I say this? What we try to encapsulate in the term private neoantigen is that a private neoantigen is representative of perhaps 99% of all neoantigens. As the name implies, a private neoantigen is restricted to individual patients. And this is because these are epitopes that are derived from a bystander process of transformation, so-called passenger mutations. Now, because passenger mutations do not directly contribute to the fitness of cancer cells, there can be both the tendency for clonal heterogeneity as well as immunologic selection pressure. A cancer cell can lose expression of a passenger mutation and still retain its lethal potential. Finally, as I was mentioning, HLA is a major complicating factor for developing TCR therapies. Because private neoantigens do not have a specific requirement for the restricting HLA allele, this can add an additional layer of complexity. By contrast, we've developed a framework which we've termed public neoantigens, and as the name encapsulates, a public neoantigen should be shared by groups of patients. Now, the reason they might be shared is because they result from recurrent genetic processes that are associated with transformation, in particular recurrent hotspot mutations in driver oncogenes, although tumor suppressor genes can also be targeted in an analogous way. Now, because driver mutations actually contribute to transformation and clonal fitness of cancer cells, the tendency for clonal heterogeneity should be far reduced, as should the tendency for immunologic selection pressure. A cancer cell cannot lose expression of a driver gene and still be a lethal process. Finally, and this is really the challenge of the matter, for a shared neoantigen to be useful, it should be restricted by an HLA allele that has some representation in the general population so as to be useful. So over the last 5 years, my lab has begun a concerted effort to identify and therapeutically target public neoantigens. This is an effort that has been led by an outstanding senior scientist in my lab, Dr. Smita Chandran, who's been joined most recently by Iñaki Extabera, a postdoctoral fellow from Spain, as well as just an outstanding group of collaborators, both at MSK as well as in external institutions. Our research strategy has coalesced around the following sequence of events. We begin by doing an immunopeptidomic screen to prove to ourselves that the combination of a recurrent mutation, either a point mutation or more recently we've evaluated recurrent gene fusions, can give rise to a shared neoantigen that is expressed by a common HLA allele. And in a couple of slides, I'll actually show you this screen in process. Now, we very intentionally do not make use of HLA in silico prediction algorithms. Although these algorithms are a great place for hypothesis generation, what we and others have found is that these in silico prediction algorithms way overestimate the existence of epitopes that are actually shown on a cancer cell surface. Perhaps 80 to 90% of all of the predicted epitopes, in fact, are not actually displayed. Now, once we've credentialed that an epitope resulting from a hotspot mutation can be presented on the surface of a cancer cell, the second step of our research process is to make use of an extraordinarily unique resource available here at MSK, termed MSK Impact. MSK Impact is a clinical next-gen sequencing platform that in some ways is analogous to other CLIA-certified commercial genetic tests such as Foundation Medicine, but with one important exception. MSK Impact is able to resolve not just the driver mutational landscape of a patient, but also the patient's unique HLA haplotype. Furthermore, we make use of a software program called Darwin, which is an electronic enrollment tool that looks ahead at our institution a couple of weeks or a couple of months in advance to look for patients that have the right mutant allele and HLA allele that gives rise to a shared neoantigen that we identified in our mass spectrometry screen. And based on that information, we are able to prospectively collect biospecimens from cancer patients who might form a shared neoantigen on the surface of their cancer cells. To date, we've collected over 170 unique biospecimens from patients who all share in common a shared neoantigen. And today I'm going to present data for about a quarter of this total population. Now, once we've developed a biorepository, we next do a series of tests to test whether this naturally processed shared epitope gives rise to a spontaneous T cell response. So we perform immune monitoring, making use of a DNA bar-coded version of a synthetic HLA molecule that has been loaded with a mass-spec identified peptide. And the reasons for doing this are twofold. One, this validates that the epitope that we discovered can generate a natural T cell response. The second is, by performing single-cell sequencing, we retrieve the unique genetic sequence that encodes a T cell receptor that can bind such epitopes. Next, we take these gene sequences, clone these into expression vectors. Our favorite expression vectors are gamma retroviruses, because this can be done in a number of different ways to compare and to contrast the different TCRs that result from a patient. And I'll tell you that this is an incredibly humbling experience because of the magic of the diversity generator of somatic recombination. Nature has found innumerable ways to solve the problem of finding a specific peptide MHC complex. And these can confer very different functional attributes and safety attributes to any individual TCR. And finally, all of this is done with a line of sight, not only to develop a new understanding of the determinants of immunogenicity and immune escape to neoantigens in general, to humans, but to develop a new class of precision TCR therapies targeting these driver oncogenes. And what we're really excited by is the fact that we are pivoting in the next year to taking some of our candidate TCRs and returning these back to patients in first-in-human clinical trials. And we will be using the exact same next-gen sequencing platform to prospectively identify patients who might benefit from such an experimental treatment. This is looking at the driver landscape of over 2,000 solid cancers from a publication by Priestley et al. In Nature just a couple of years ago. What I like about this data set is that, unlike TCGA, which many of you may be familiar with, this particular data set looked exclusively at the genomes of metastatic solid cancers. TCGA is disproportionately weighted to localized cancers. And of course, it is metastatic disease that by and large contributes to cancer-related deaths. What you can see is that in terms of frequency, the most commonly mutated driver oncogenes include PI3 kinase at the top, as well as a number of different drivers that may be familiar to you all, including KRAS, BRAF, and as I'll talk about a little later, NRAS. Zooming in, if you look at the precise genomic alterations that are associated with mutant PI3 kinase, you can see that the vast majority are missense mutations that result in a codon change or an amino acid change in the resulting protein. And if you zoom into the gene body, and this is really typical of most drivers, mutated drivers, you can see that mutations occur not throughout the gene body, but in very tightly constrained hotspot regions. In the case of PI3 kinase, the vast majority of the mutations occur here in the kinase domain at the H1047 position, with the substitution of either an arginine or leucine in place of the native histidine. So here is our mass spectrometry screen in action. We begin by making use of COS7 cells. So COS7 is a monkey-derived cell line that has antigen processing genes that are nearly identical to that of humans. But of course, a monkey-derived cell line does not express naturally human HLA alleles. Into COS7, we electroporate the full-length gene sequence that encodes a driver of interest, in this case, all of the major hotspot substitutions for PI3 kinase, including wild-type PI3 kinase as a control. And in a tile fashion, we co-electroporate in genes encoding the most prevalent HLA alleles in North America as resolved by MSK impact for both the A, B, and C alleles. We then do immune precipitation mass spectrometry to ask a simple question. Is there a combination of one of these driver hotspot substitutions and a common HLA allele that gives rise to a naturally processed and presented public neoantigen? This is a representative mass spectrometry screening result that shows actually the answer is yes. So here we can see that a mutated form of PI3 kinase, in this case, the H1047L substitution, the second most prevalent amino acid substitution at the most common hotspot position of PI3 kinase, gives rise to an epitope that is restricted by the highly prevalent HLA-A3 allele. Now, when we did the exact same experiment with a couple of different controls, it gave some important clues into the mechanism of the epitope formation. First of all, in cells that express wild-type PI3 kinase and HLA-A3, we did not detect an epitope, a finding I'll come back to shortly. And as a specificity control, if we mismatch the HLA allele, here A69, but have mutant PI3 kinase, we also could not see evidence of an epitope. We resolved that the epitope in this case is a 9-amino acid residue, the sequence shown up here, in which the hotspot substitution, a histidine for a leucine residue, occurred in the second position. Now, it is often the case that for HLA class 1 molecules, the second amino acid and the final amino acid are so-called HLA anchor residues. These are residues that point down from the peptide and physically engage the HLA molecule to stabilize the peptide. Based on the location of where the hotspot mutation is in this peptide, combined with the fact that we could not see the wild-type form of the peptide naturally processed and presented, we hypothesized that the hotspot substitution created a favorable and stabilizing HLA anchor residue. To formally test this possibility, we performed molecular anisotropy here at physiologic temperatures and found that indeed, consistent with our hypothesis, the mutated peptide has a prolonged half-life measured in many, many hours, whereas the wild-type form of the peptide fell apart before our eyes, and in fact, we almost could not experimentally measure a half-life with the wild-type peptide. Now, having credentialed the existence of a shared neoantigen resulting from mutant PI3 kinase, we subsequently did additional genomic studies using multi-regional biopsies from patients who expressed the PI3 kinase shared neoantigen, and in multiple subjects, including subjects with breast cancer, a cancer type that is highly enriched in PIK3CA mutations, as well as endometrial cancer, another cancer type that is also enriched in PI3 kinase mutations. We could see that the PI3 kinase mutation is clonally conserved across every metastatic site in addition to the primary, whereas there is passenger mutations that form along the branches. Now, having established the existence of a clonally conserved shared neoantigen from PI3 kinase, we next ask the question, can this give rise to a spontaneous T-cell response in patients? Again, we made use of MSK-IMPACT to collect a cohort of 14 unique patients who all have different cancer types, different genders, different cancer treatment histories, but all share in common at least one copy of HLA-A3 and the mutant PI3 kinase substitution that can give rise to the shared neoantigen. Here are some representative immune monitoring results from these studies. This is an HLA-A3 healthy donor where we stain the cells with synthetic peptide HLA complexes that are fluorescently labeled and loaded with the mass-spec identified shared neoantigen. In no healthy donor could we find evidence of circulating clonally expanded T cells that could recognize this epitope. But in perhaps a third of cancer patients with diverse cancer types, including breast cancer, anaplastic thyroid cancer, which is an extremely aggressive form of thyroid cancer, and bladder cancer, we could find that patients could mount a spontaneous T cell response to these epitopes, demonstrating that this is in fact immunogenic. We have subsequently gone on to clone a panel or library of T cell receptor gene sequences from these patients as well as from healthy donors and compared and contrast their function. This is intracellular flow cytometry of a combination of CD8 and CD4 positive T cells that have been transduced with the TCR gene sequences encoding these library members. This is looking at T cell function in response to target cells that express wild-type PI3 kinase and HLA-A3. And what you can see is that all of these TCRs have a very clean off state. They do not respond to normal cells that express wild-type PI3 kinase. However, when co-cultured in the presence of cancer cells that express the hotspot mutation, all of these TCRs become activated, here as evidenced by TNF-alpha secretion, although the T cells mount a multivalent response, secreting other cytokines, including interferon gamma, as well as acquiring cytolytic capacity in a potent and specific fashion. Now, despite the fact that all of these TCRs share in common PI3 kinase hotspot specific function, they do differ in a couple of important ways. What I want to draw your attention to is that we very intentionally transduced a population of CD8 positive T cells, shown here in red, as well as CD4 positive T cells or CD8 negative cells here in gray. Although all of the TCRs could trigger in CD8 positive T cells, it was only a rare subset that could also trigger in CD4 positive T cells, as shown by the green arrows in the case of TCR3 and 4, but not in these other TCR candidates, TCR1 and 2. Well, what does this mean? When you express a TCR that recognizes a class 1 restricted epitope in CD8 positive T cells, the CD8 co-receptor engages an invariant region of the HLA molecule that increases the avidity of this interaction. If you take the same TCR and transduce it into CD4 positive T cells, because the CD4 co-receptor cannot bind the HLA molecule, the extra avidity push of this interaction cannot take place. In other words, this is a really simple way of assessing the intrinsic affinity for the TCR for the peptide MHC complex, and implies that TCR3 and 4 may have a particularly high affinity for this peptide MHC complex compared with TCR1 and 2. This is looking at real-time cytolytic function of each of these TCRs. Here you can see that all of the TCRs have minimal to no background cytolytic activity against healthy tissues that express HLA-A3 and wild-type PI3 kinase. And all of our TCRs mediate mutation-specific cytolytic function. And in particular, the two TCRs that were co-receptor independent, TCR4 and 3, across multiple experiments, mediated the most profound cytolytic capacity. Now, although TCR4 and 3 share potent cytolytic activity, co-receptor independence, they are also distinguishable from one another. And one way in which they are distinguished is that we notice that TCR4 has a profoundly long CDR3 beta loop length compared with TCR3, as well as a panel of over 17,000 HLA-A3 or A11 restricted TCRs. What is CDR3? There are two different highly polymorphic regions of a TCR, the CDR3 alpha and beta loops. These are the loops that actually physically project from the end of a TCR and physically come in contact with the peptide MHC complex. We next ask the question, what contribution, if any, does this inordinately long CDR3 beta loop for TCR4 contribute to this particular TCR's function and potency? To address this question, we teamed up with Brian Baker, who is the former chair of structural biology at the University of Notre Dame, and did X-ray crystallography of both of these TCRs in complex with the shared neoantigen bound by HLA-A3. Now, this is honest to goodness X-ray crystallography. You can download these from the protein database. This is not an AlphaFold in silico predicted study. What we found is that TCR3 binds in what is a very canonical or average way for a TCR. It does not bind the entire peptide, but rather focuses its attention on the central core region of the peptide. And here you can see the CDR3 alpha and beta loops coming down and physically touching the peptide. What I hope is apparent to you is that TCR4, with its inordinately long CDR3 beta loop, has an extended loop that can form a curtain over the vast majority of the peptide. It's sensing a lot more of the neoantigen than TCR3. So let's zoom in and take an even deeper look at this interaction. Here you can see selected residues from TCR4, and you can see that the vast majority of the HLA-bound peptide is physically engaged. Here I'm showing hydrogen bonds, but also van der Waals interactions and other interactions are engaged by the TCR loops with the peptide. Now, beyond being a biologic curiosity and a pretty picture, this actually has extremely important clinical and translational implications. This is what is called alanine glycine scanning mutagenesis, and this is a way of inferring the potential for cross-reactivity of a TCR. In this experiment, we take the native peptide gene, amino acid sequence, and at each position, mutate that position for an alanine, a glycine residue, or any of the 20 standard proteogenic amino acids to ask the question, is the TCR tolerant of these changes or intolerant of these changes? And what we found, and really I think dovetails nicely with our structural data, is that if we mutated 8 of the 9 native amino acid positions, the TCR completely loses function, with the exception of position 1, which is right here, and you can see that the TCR does not physically engage this residue. This has a very important upshot. This means that experimentally, this TCR is incapable of recognizing and meeting off-target effects in any human peptide. Now, to assess preclinically the potential therapeutic implications of this TCR, we took TCR5 and used it in a treatment of 2 isogenic tumor cell lines that express either mutant PI3 kinase, or as I'll show you, wild-type PI3 kinase, using an HLA-A3 restricted influenza TCR as a control. What you can see is that only mice that received TCR4 saw evidence of tumor regression and enhanced overall survival compared with the flu-specific TCR. And as a further demonstration of safety, we found that TCR4 mediated no anti-tumor effect on tumors that express HLA-A3, but wild-type PI3 kinase. So in conclusion for this first part, we found that a recurrent mutation in PI3 kinase, again, one of the most prevalent driver mutations in human oncology, creates a shared immunogenic public neoantigen. The molecular basis for immunogenicity in this case is that the hotspot substitution introduces a preferred stabilizing HLA anchor residue that allows an epitope to be stably presented on the surface of cancer cells, which can drive a T-cell response. The wild-type form of the peptide, by contrast, has an unfavorable residue in the second position, resulting in an unstable complex, which gives rise to a very, very nice safety threshold for a TCR therapeutic. Further, I've shown you an example of where we've identified a rare TCR, which is now our lead TCR candidate, which we are seeking to begin a first-in-human study in early next year, that is mutation-specific, lacks off-target reactivity, is effective in vivo and preclinical models, and a lot of these functional attributes are due to an inordinately long, uniquely long CDR3 beta loop that can fully engage the peptide. So I now want to pivot to unpublished data regarding our efforts to target mutant NRAS. So mutant NRAS is a member of the RAS family of driver oncogenes. RAS is mutated in approximately 1 cancer in 20, and NRAS is the second most common RAS mutant isoform besides KRAS. So why focus on NRAS? Well, there's a couple of different biologic regions. One is there's extraordinary unmet medical need for mutant NRAS cancers. Melanoma has appropriately been viewed as a great bellwether of the success of immunotherapy. Nevertheless, it's the case that not all molecular subtypes of melanoma are equally responsive to current anti-PD-1, PD-L1 agents. And in point of fact, in data recently published by my colleague Alex Shustari, patients who have either mutant NRAS or mutant BRAF melanoma cancers disproportionately respond poorly and have a shorter time to treatment failure compared with patients with wild type or mutant NF1 melanoma. However, unlike mutant BRAF, there are currently no small molecule inhibitors for mutant NRAS, identifying mutant NRAS melanoma as an area of key unmet medical need. Similarly, about 7% of all colorectal cancers are associated with mutant NRAS. Like mutant KRAS, mutant NRAS is a biomarker for therapeutic resistance to current anti-EGFR therapies. And in point of fact, in data from my colleague Andrea Sursic here at MSK, patients with mutant NRAS have an even more aggressive course, impaired overall survival compared with even mutant KRAS cancers. Again, identifying another prevalent disease with high unmet medical need. The vast majority of hotspot substitutions in mutant NRAS are focused along the Q61 codon with 2 basic amino acids, either a substitution of an arginine or a lysine, accounting for over 90% of the hotspot substitutions at this position, followed by a distant third, substitution of a leucine residue. As you can see in this crystal structure, the Q61 residue is located right here in the active site of NRAS. This substitution locks the protein in a constitutively active GTB-bound state because it cannot hydrolyze this GTP to GDP, resulting in transformation. As we did with PI3 kinase, we again began with our mass spectrometry screen, electroporating in the most common hotspot substitutions for NRAS Q61 using wild-type NRAS as a control, and again, the most prevalent HLA alleles here at MSK. Our mass spectrometry results found that in point of fact, there are a series of shared or public neoantigens that result from all of the prevalent NRAS Q61 hotspot substitutions. This is because the NRAS peptide has ideal HLA anchors already to bind to HLA-A1, the second most common HLA allele in North America. And you can see that all of the hotspot substitutions occur here in the center of the peptide. Really a nice contrast or corollary with what I showed you with PI3 kinase. Now, because NRAS already has ideal HLA anchor residues for HLA-A1, it is also the case that wild-type NRAS is also naturally processed and presented on all nucleated cells that have HLA-A1. So again, I want to emphasize that unlike mutant PI3 kinase, which I showed you, here is an example where the hotspot introduces not a stabilizing HLA residue, but a residue that is in the center of the peptide and presumably pointing up into the solvent up towards where a TCR might engage. Based on these questions, we addressed 3 different experimental questions. Because mutant NRAS as well as wild-type NRAS is expressed on the surface of HLA-A1 positive cells, this implies that wild-type NRAS is expressed within the thymic epithelium. Therefore, our first question was, does thymic epithelial expression, which may cause central tolerance to NRAS, limit the ability to form an immunogenic response against mutant NRAS? And furthermore, does this, if we are able to see a T cell response, does this compromise T cell functional avidity? Second of all, exploiting the fact that the hotspot substitutions are either basic or aliphatic, we next address the question, does the chemical nature of the hotspot substitution alter the tendency for public neoantigen immunogenicity? And finally, can we use TCRs that are cloned from patients as a form of TCR therapy, analogous to mutant PI3 kinase, for a new class of therapies targeting mutant NRAS cancers? To address these questions, we again made use of MSK Impact, where we curated a biotrust of patients who have in common HLA-A1 and either the Q61R, K, or L hotspot substitution. In this case, we were able to get biospecimens for over 34 subjects, including not only peripheral blood, but in selected cases also tumor-infiltrating lymphocytes from diverse cancers that are commonly associated with mutant NRAS, including melanoma, colon cancer, and thyroid cancer. Here is the results of our initial immunogenicity screens, efforts that have been led by Iñaki Extabera in my laboratory. So here we have 12 different subjects, again, diverse cancer types, different treatment histories, but all have in common at least one copy of HLA-A1 and a tumor that expresses NRAS Q61R. In this case, we found that about 2/3 of patients, or 60%, have evidence of a spontaneous T cell response. Here is dual dextramer staining with the mass spec identified either mutant peptide containing Q61R or wild-type NRAS peptide. You can see that patients with diverse cancer types, not just melanoma, but as well as cancer types that are normally thought to be poorly immunogenic, such as germ cell tumors or mismatch repair proficient, sort of the poorly immunogenic colon adenocarcinoma, can all have a detectable T cell response against the Q61R hotspot. And all of these T cell responses are mutation specific because these same T cells do not bind the wild type version of the peptide. And finally, HLA-A1 positive healthy donors also do not mount a spontaneous T cell response. So how about the other NRAS hotspot substitutions? Here is the second basic amino acid substitution, the NRAS Q61K. And here, in a cohort of 9 patients, we came to a very similar conclusion. The patients with melanoma, as well as mismatch repair proficient colon adenocarcinoma, can generate a spontaneous T cell response against this epitope that is entirely mutation specific. And finally, here are a group of patients who have the NRAS Q61L, third most prevalent NRAS hotspot substitution. And again, about 60% of patients develop a spontaneous T cell response detectable in the peripheral blood that is mutation specific. So this implies that the chemical nature of the hotspot mutation really has no bearing on the ability to detect a T cell response, nor does thymic epithelial expression of the wild type version of NRAS. Now, all of these experiments were done in the peripheral blood. Can the T cells make their way to the tumor? So in a subset of patients, such as this 50-year-old patient with metastatic melanoma, we had access to not only peripheral blood, but as well as TIL. And what you can see is that this T cell response against the NRAS hotspot mutation can be found in multiple compartments in the patient. They are not just restricted to the peripheral blood. They can also make their way to the tumor site. We have found that there appears to be a correlation between the detection in melanoma patients of these T cells and the likelihood of response to immune checkpoint therapy. Here I show a 65-year-old patient with metastatic melanoma, widely metastatic, including to the brain, lung, adrenal gland, and skin, who received combination immune checkpoint inhibitors as part of standard of care by my colleague Mike Postel, who's had a very deep and prolonged partial response now lasting over 16 months. You can see this very large adrenal mass that has nearly completely regressed, as well as this cutaneous metastases in this patient that has also regressed. And in the circulation of this patient, perhaps one T cell in 100 can recognize this shared neoantigen. So in total, as I was mentioning, across each of the patients we've looked at, regardless of the chemical nature of the hotspot mutation, and regardless of the location of where we looked for immune cells, about 50% of patients mount a spontaneous or detectable T cell response against this family of shared neoantigens. We've gone on then to clone the unique TCR gene sequences that are associated with these responses, again, taking patient peripheral blood that has a detectable T cell response, incubating it with an HLA multimer that has been barcoded with a DNA barcode, performing single cell sequencing to resolve the specific TCR clonotypes that are associated with binding to this molecular barcode, and then cloning these TCRs into expression vectors. Now, using this information, we can address a couple of different questions. One is to assess the question of what are the clonal dynamics of T cell responses to the shared neoantigens in cancer patients who have received immunotherapy. And here with Mike Postel, we had a very unique set of peripheral blood from patients with melanoma who received combination anti-CTLA-4, anti-PD-1 at baseline before treatment and serially after treatment. And in multiple patients, consistent with the other patient that I showed you, we found that there was either a de novo response of NRAS-specific T cells or an increase in the NRAS reactive cells as a function of time on being on immunotherapy in patients who had a good clinical response to the immunotherapy. Moreover, when we look at the unique TCR gene sequences, we can see that across these patients, there is a polyclonal response, meaning multiple TCR clonotypes recognizing the NRAS mutation are induced and expand in the peripheral blood. And as a control, we've looked at viral-specific TCRs, including TCRs against the chronic virus CMV, and found that the homeostasis of these viral-specific T cells is not influenced by anti-PD-1, anti-PD-1. We've cloned these TCRs and expressed them in both CD8 and CD4 positive T cells, just as we did with PI3 kinase, to address the question, are these TCRs cancer specific and cancer mutation selective? This is looking at a panel of TCRs that we cloned from a single patient in response to HLA-A1 positive melanoma cell lines that either express wild type NRAS as a control or the NRAS Q61R mutation. As a negative control in these experiments are untransduced T cells. And what you can see is that all of these patient-derived TCRs are capable of recognizing naturally processed and presented hotspot-containing NRAS epitope, but do not respond to wild-type NRAS. Furthermore, when we transduce the TCR into CD4 positive T cells, which again do not have benefit of the CD8 co-receptor to enhance the avidity of the interaction, we also found that all of our patient-derived TCRs are co-receptor independent, a marker of high functional avidity. Now, what was surprising to us is that we included as a control melanoma cell lines that express HLA-A1, but harbor the alternative hotspot mutation, in this case, the NRAS Q61K mutation. And to our surprise, we found that a subset of our TCRs recognized not just the Q61R expressing tumor cell line, but also the Q61K cell line. So these TCRs appeared to have a degree of cross-reactivity, or what we're terming cross-protection, to multiple hotspot mutations. Additionally, this feature of being able to recognize multiple hotspot mutations appeared to be CD8 dependent, because in CD4 positive T cells, although they can recognize the Q61R mutation, they did not respond to the tumor cell line expressing Q61K. So what is the physiologic significance of this finding? Here's an experiment in which we transduced T cells as a control with an HLA-A1 restricted TCR that recognizes an epitope derived from SARS-CoV-2 with two different melanoma cell lines that have been labeled with two different fluorescent dyes, either Q61R in green or Q61K in red. And what you can see is that a more or less equal clonal expansion of the green and the red tumor cell lines with a control TCR. TCR1, which has this ability to recognize multiple hotspot mutations, what we're terming cross-protection, could clear both tumor cell lines, whereas 2CR11, which was Q61R specific, could clear the green tumor cell lines, but left in place Q61K. These findings give an indication that this is actually a physiologically relevant finding. To further extend this, here we're looking at a time-resolved tumor growth kinetics, again against wild-type NRAS expressing tumors. Neither the control TCR or either of the two NRAS TCRs provided any protection. In response to a tumor cell line that has Q61R, the control TCR does not control tumor growth, but both TCR1 and 11 do. But it is with the NRAS Q61K that the cross-protective TCR can also mediate tumor control, whereas TCR11, which is not cross-protective, can only recognize the Q61R mutation, is unable to control. Finally, I want to return to the observation that although these TCRs, when transduced into CD4 positive T cells, can mediate activation and cytokine release, but not cytolysis, to interrogate this a little further, we did an experiment in which we transduced CD4 positive T cells with an inert transmembrane protein truncated EGFR control. This is looking at two different tumor cell lines, R and K, of CD4 positive T cells transduced with TCR1, which is cross-protective, and the inert EGFR truncated protein. You can see no cytolysis is mediated by these CD4s. Therefore, we transduced in exogenous CD8 into these CD4 positive T cells, and we found that this turns the helper CD4 into a killer CD4 just by transducing in CD8 co-receptor, and it allows the co-receptor to function in a way that provides cross-protection to both the two NRAS hotspot substitutions. So to conclude this second part of the talk, we have found that all NRAS Q61 hotspot mutations, regardless of the chemical nature of the hotspot substitution, and regardless of the fact that the wild-type version of NRAS is expressed in the thymic epithelium, is immunogenic in the context of HLA-A1 in a diverse set of cancer patients. Furthermore, we have found a correlation in patients with the Q61R substitution that these T cells are responsive to combination immunotherapy in vivo and appear to be associated, at least in some subjects, with objective tumor regression. Finally, we found that a panel of TCRs have a unique property. It really is cross-reactivity, but in this